Hello, I hope that you all had a great Halloween and that you are eating all of your Halloween candy in moderation. Please remember that you have an exam next Thursday on D2L. If you do not let me know before the exam that you cannot take the exam, or if you miss it because you think it will be on Friday, you will be able to retake it, but there will be a significant reduction in your grade. So please pay attention to the fact that it is going to be on Thursday and not Friday, and this will be next week. Stay tuned for an exam study guide which will be posted by early next week. A while back I had said that there would be a Unit 3 quiz, which would be this week, but that is no longer taking place, and there will be no Unit 4 quiz as well, so we just have the exam next week. With that out of the way, we are on our way to Chapter 29. We are going to cover all of the material in this chapter in this video, but we will really hone in on the freezing and canning when we go over this chapter the week of November 30th. We are going to break up this chapter into two major sections. The first main focus for this chapter will be on different things that can influence foods to make them unsafe for us, and then we'll follow that up by discussing ways that we can prevent these problems from occurring. With each of these learning goals, what you are really trying to determine is what causes food spoilage and what can we do to prevent food spoilage. To understand that, be able to identify environmental and storage conditions that commonly pose risks for unsafe foods. Be able to correlate how exposure to microorganisms, enzymes, bruising, and oxidation affect spoilage rates of certain foods. Explain the mechanism for how moisture control, temperature control, and irradiation work to prevent food spoilage, and finally be able to explain the main goals of packaging and discuss how packaging delays food spoilage. Earlier we had mentioned that this chapter can be broken down into two major sections. Now there are three different bullet points here. The first one talks about that first section. So what causes foods to go bad, what causes them to spoil and then to become contaminated. And then the second and third sections of this chapter will look at that second major section of this chapter and talk about what we can do to prevent foods from going bad. So to do that, we're going to look at what we do with temperature and moisture control. And then we'll talk about what irradiation is and how that keeps foods fresh. And then finally, we'll briefly discuss packaging, what that is, what some different techniques are, and then how packaging can prevent foods from going bad. You do not necessarily have to memorize these terms for the exam. Some of these may be on here, but the important thing to understand is if you know what the key terms are, if you understand what these definitions are, it's going to make the chapter much simpler. So before you go through the chapter, take a minute to review these and have a general understanding of what each of these terms are. Sterilization and pasteurization are often used hand in hand, but they are different things. Both involve heating the food to kill microorganisms, but with pasteurization there's a lower amount of heat applied, so not all of the microorganisms are killed, just most of them. With sterilization, you are killing all of the microorganisms, so there's a higher heat that's applied to sterilization compared to pasteurization. We've talked about the temperature danger zone. Be familiar with this. This is 40 to 140 degrees Fahrenheit, and during this temperature range, microorganisms, they can live and they can also multiply. Irradiation, we'll talk a little bit more about this in future slides in this chapter, but that is the application of X-rays to kill microorganisms in a food. This first part of the lecture looks at what causes foods to go bad. Food spoilage is caused by exposure to external factors. We know that bacteria will cause food to go bad, and we have reviewed the effect of oxygen on food spoilage. And it is obvious that adding non-food objects and chemicals not meant to be in the food will also be bad for the food. But what about bruising and enzymes? We will talk more about all of these on slides to come. For the contaminants, let's start off with microorganisms and bad bacteria. There are millions, if not billions, of types of bacteria. Some are good and some are bad. A healthy digestive system is dependent on a healthy balance of bacteria, as the bacteria in our large intestine helps to further break down and digest our food. We eat yogurts because yogurt contains probiotics, which is actually bacteria that's going to support the healthy functioning of our digestive system. Bacteria can go through fermentation, during which sugar is eaten by the bacteria, or in some cases an enzyme, and that produces a byproduct that creates a desirable reaction, whatever that may be. Remember how cheese and yogurt are produced? For both of these, the goal is to separate the casein, which is done through fermentation by adding bacteria. The flavor of sauerkraut and pickles is strongly linked to fermentation from adding certain bacteria. Unfortunately, there are also plenty of bad bacteria that are related to potential disease that we can develop if we consume a high enough level of these microorganisms. In most cases, you will not be able to visually see microorganism contamination, and the food you are eating will look perfectly fine. Salmonella, E. coli, and Listeria are just a few examples of foodborne illnesses that can lead to severe complications and in some cases, death. Just like us, bacteria are very picky and have to have things a certain way in order to survive and also to thrive. And this is when bacterial contamination can occur, when they are thriving. Bacteria will multiply, and the closer the environmental conditions are to what is optimal for them, the faster they will multiply, making them more likely to cause contamination in our bodies once we eat these foods. So they require a few different conditions to be present. The first two are obvious: food and the right acidity. There is no organism that can survive without these two things. The next four are pretty obvious, but this is largely what we will pay attention to when controlling conditions to prevent spoilage and contamination. So pay very close attention to these. Time and temperature. You already know this one from the danger zone. Remember the time frame surrounding the danger zone. Bacteria is going to die at high temperatures and hibernate at low ones. And the longer the time spent between this range, the more the bacteria can multiply. More on this later. Oxygen is the next one. Bacteria needs air. It makes sense. And finally, moisture. Bacteria must be hydrated. Moisture is simply water or liquid which can be found in nearly every single food. If you put this all together, bacteria are very similar to humans. We all require all of these things. If we don't have any food, we starve. If our acidity is just slightly out of balance, we die. Too much hot or too cold, we overheat or we freeze. Without oxygen, we suffocate. And without moisture or water, we will dehydrate and wilt away. Enzymes are simply forms of protein. They basically just make chemical reactions occur and often speed up their process. Spoilage is usually little more than a series of chemical reactions that occur, which can be easily traced back in some way to enzyme activity. Oxidative enzymatic browning, which you can see in both of the pictures on the top of the screen, is a process that occurs when the insides of certain fruits and vegetables are exposed to oxygen, which is going to trigger an enzymatic reaction that causes the browning. This is one form of food spoilage, but it can present itself in a number of other ways. Physical and chemical contaminants are pretty obvious things that should not be in food. A physical contaminant could be any object such as glass, hair, nails, And this object by itself, it could cause damage when you eat this. The object also could be carrying microorganisms and bad bacteria that could cause another type of spoilage or contamination of the food. Chemical objects are cleaning products or other chemicals that are typically found in a kitchen setting that are not intended for food that are, which is usually accidental, added to a food. To give you an idea of what a physical contaminant is, I'm going to share just a brief story. One day I was out grocery shopping and bought a package of muffin bread. I was quite hungry, so on the car ride home, I opened this package and I started to eat this bread. On one of the bites, I could tell that something was way off. So I spit it out, looked down, and right in the middle of the bread was a very large rusted screw. The screw is a great example of a physical contaminant. Although it does carry bacteria on it, by itself it is a physical object that can cause damage to the consumer. So nails, hair, a broken off tip of a pencil or a marble are all examples of physical contaminants that packaging typically bars from coming into contact with a food item after the food is packaged. We're going to break this video up with a quick critical thinking question. Now think about this for a second. This should be easy for you. If olive oil is left outside of its container for 24 hours at 80 degrees Fahrenheit and the olive oil goes bad, what caused the olive oil to go bad? So let's think about this. The olive oil is left outside of its container for a long period of time, smack dab in the middle of the danger zone with exposure to plenty of oxygen. What was the cause of the contamination? So it was oxidation and not bacteria. Can you guess why it was not bacteria? Think about olive oil, think about its chemical properties. Olive oil is just fat. How much water is in olive oil? How much liquid is in olive oil? Well, olive oil is a liquid, but the amount of water or moisture is non-existent. There is no water, no moisture, and therefore bacteria cannot grow. Exposure to oxygen is another way that the quality of a food item can be worsened. Fruits and vegetables have outer layers of skin that do not negatively interact with oxygen, while the insides of many fruits and vegetables do have this negative reaction that causes them to go bad. In these cases, the skin of the fruits and vegetables is a barrier for many things, one of those being chemical damage that can occur due to exposure to oxygen. Consider enzymatic oxidative damage that occurs to an apple after you take a bite and let the apple sit out for some time. This is due to exposure to oxygen or to oxidation. Leaving the olive oil outside of its container in the presence of oxygen turns it bad and leads to rancidity. Bruising increases the susceptibility of food to infection. So whenever food is bruised, there's cellular damage that's occurring, and there may be also oxygen that's being let into the food that causes it to go bad. Either way, the food becomes more susceptible to microorganism contamination when it is bruised, and the rate of spoilage from bruising due to the cellular damage is going to be increased. So that's all the bad stuff can happen. Now we're going to look at what we can do to get foods away from contamination risks. So in section 2, we're looking at temperature control, we're looking at controlling the moisture or water content of foods, and then we're also going to be talking about irradiation and how that process keeps foods fresh for longer. Remember that moisture and temperature are both required for the growth of microorganisms. If we control both of these factors correctly, then bacteria cannot grow. or it at least cannot do so at a very rapid rate. For temperature control, hot temperatures kill bacteria, while cold temperatures don't necessarily kill bacteria, but they do prevent them from multiplying. For moisture control, by limiting the water or moisture, bacteria will not be able to grow. Food service has a variety of practices in place to ensure both temperature and moisture control, and we will cover a few of these in the next few slides. When going over this material, keep in mind that you have already learned quite a bit about the temperature danger zone and what goes on when food and bacteria are left to grow in that range. For irradiation, by sending rays into foods, irradiation kills bacteria and potential pathogens. You already know this and we've already covered it, but we will review it one more time. Leaving food out at any temperatures between 40 and 140 degrees will allow bacteria that is inside of food to grow and multiply to potentially dangerous levels. Bacteria can simply not live at a temperature above 140 degrees. Above 140 degrees, bacteria will die. Below 32 degrees, the bacteria freezes solid and goes into hibernation. Note that hibernation is quite a bit different than dying. At these temperatures, bacteria simply become inactive and can no longer grow. However, once the food is reheated, or when it thaws, the bacteria will start off right where it left off and begin to multiply again. Between 33 and 40 degrees, which is temperatures for refrigeration, bacteria can still grow and multiply, but their rate of multiplication is very low, so low that food can be refrigerated safely at these temperatures for a relatively long period of time. So ultimately, our goal with temperature control is either to heat foods to kill bacteria or put foods in cold temperatures to prevent microbial contamination. The first two forms of temperature control are very simple. We're talking about things that even you and I can do. We do not have to have heavy equipment and machinery to refrigerate and freeze foods. In both of these cases, we are just trying to slow down bacteria growth either considerably with refrigeration or to a complete halt with freezing. So refrigeration is storing foods between 33 and 40 degrees Fahrenheit. Freezing is holding temperatures below 32 degrees Fahrenheit. The next two processes are more commonly found in large food service establishments, and these are pasteurization and canning. Pasteurization involves heating foods to a temperature that kills the vast majority of microorganisms, but does not kill all of them. When foods are pasteurized in a container, microbial contamination will be delayed for a very long period of time. This is different from sterilization, as sterilization involves heating foods to a higher temperature than pasteurization, which will kill all of the microorganisms. Canning, we're going to discuss more about this in the last unit of class, but we will highlight the basic principles of canning here. Canning is a bit more than temperature control. With canning, we are also sealing off the food from oxygen. Controlling both oxygen and temperature allows the shelf life of these foods to be incredibly long. And usually when we put foods into cans, we have already killed a significant amount of the bacteria, so there's not much bacteria already present in cans. And sometimes canned foods are completely sterilized, and complete sterilization, that means the presence of no bacteria in a very neutral environment. Moisture is in some way, shape, or form related to the presence of water, and bacteria requires water or moisture to survive and to multiply. Water can be found inside of foods and also in the air of very humid environments. So somebody living in the desert of Arizona, when they have a piece of a fruit or a vegetable, because it's so dry there, that vegetable or fruit is much less likely to spoil from bacterial contamination because there's much lower chance of that food from being excessively moist. So it's not a very ripe environment for bacterial growth. Meanwhile, if you are in Charleston right after a very large series of thunderstorms where it's been raining for a few days, the moisture content is going to be very high. It's going to be a very high level of humidity in that air, and fruits and vegetables in Charleston will be much more likely to spoil due to microbial contamination, due to the presence of water in the air itself there. Moisture control is more so about moisture reduction. For foods that have extended shelf lives due to moisture control, their water content is decreased through some form of evaporation. With low enough water content, generally somewhere below 30% or less of the food's composition being water, with that amount, the bacteria will be dehydrated and unable to survive. The moisture control process is mainly performed through either drying or freeze drying. To dry a food, you simply apply heat. So this could be done through the sun, or sun drying, or through the application of other heat waves to the point where the water is going to just evaporate from the food. Very simple. For freeze drying, you freeze the food and then you heat this in with very low and very concentrated amounts of heat. So can you think of an advantage of this? The majority of the food is frozen and you're heating little bits of it and then dehydrating it as you heat those little bits. So if the food stays frozen, can bacteria and microorganisms grow? No, because at this temperature, at a frozen level, bacteria and microorganisms go into hibernation. Now there is deterioration of foods that can occur when temperatures are warm but below 140 degrees. The freeze drying method prevents these harmful changes from occurring, which will also help the food to retain a higher level of flavor, and the structure is less likely to be damaged. So most of the food remains frozen and then you're dehydrating little pieces of it. So the frozen food cannot grow bacteria. The food that's warmed is dried, and so the moisture content is very low, and because it's so low, bacteria cannot survive in that. So even though that food is within the temperature danger zone, it can still not promote the life of bacteria to the point where it can thrive. There might be a little bit of bacteria, but it's going to be very low amounts because the moisture level is so low. Now we could also reduce moisture through osmosis. If you can correctly explain how this process works, you will be eligible for 2 extra credit points. I will give you one hint, and that's craisins. If you can explain this, please send me an email with your section number in the header, and I will make sure that you get these points. Irradiation is somewhat similar to microwaving, in the sense that with irradiation and microwaving, both of these involve the application of microscopic rays to the food, and there is no actual heat that's applied to the food. Now in microwaving, the food is heated, but that's not going on during irradiation. In irradiation, rays are sent directly into the food, which is going to break apart the DNA of the microorganisms and kill the microorganisms. Now this is a very interesting process because fortunately for us, the microorganism structure is much weaker than the molecular structure of the food. So while these rays are plenty powerful to kill the microorganisms, they are not going to have any significant detrimental effect on the food. So that's going to kill the harmful microorganisms, but it's going to keep in the nutrients, maintain the texture and flavor of the foods that irradiation is applied to. Adding preservatives is going to increase the shelf life of foods or preserve foods for a much longer period of time. And preservatives do this through various methods. Acid is going to decrease the pH of foods so that bacteria cannot survive. Sugar binds to the water in the food, making it less accessible for microorganisms, making it a drier product for microorganisms, so they'd be more likely to dehydrate if they were placed in a food with a higher sugar content. Antioxidants, you can pretty much break this one down. Antioxidants prevent damage that can occur from exposure of the food to oxygen. Salt draws water out of food, so microorganisms do not have sufficient moisture. And there are many other preservatives, and they have a variety of different mechanisms that are used to extend the shelf life of the foods that they're applied to. Now, preserving the shelf life of foods can come at a cost. Today we have a much better control on the types of preservatives allowed in our food supply, and any food items or preservatives that have a strong link to chronic disease are not allowed in our foods. But before, there was a lot less regulation of this, and there were a lot of things in foods that were very, very harmful. So many years ago, the United States came up with what's known as the Poison Squad to investigate the food additives that were being put in certain foods that were sent to grocery stores, and families would eat these foods and believe that they had no risk to them. And they had very strong chemicals in them to keep foods fresher for longer. Again, keep in mind that even the preservatives today, with the regulations that we do have some links to serious health consequences. But with the Poison Squad, there was much less regulation. So these guys would be given foods to eat, and they volunteered to do so. They were given foods to eat that had these preservatives that may have been strongly linked to health consequences. And so they would eat these foods, and many of them became very, very sick and very, very ill. And through that process, they were able to learn that a lot of preservatives needed to be X'd and nixed from the food system and completely cut out from the consumer supply. So the ones that we have today, again, they're much better than the ones from the early 1900s when the Poison Squad was around and needed to be in place to determine whether certain additives were bad for foods. But the ones that are in our system today, things like sugar. Sugar can cause inflammation, and if it is too high, can lead to unstable energy levels. Salt can increase our blood pressure. Trans fat is related strongly to inflammation, heart disease, cancer, diabetes, and many other chronic health conditions. Unfortunately, it will be phased out in 2018. And then nitrates, those are preservatives that are linked to inflammation and cancers. So we're going to finish up by highlighting some of the main concepts of food packaging. We're not going to go too in depth with this topic, but we'll at least highlight the main points and talk about some more intensive methods of packaging that certain food manufacturers will do to ensure that the shelf life of their foods is extended for a much longer period of time. And a lot of this is going to go back to the same principles that we've talked about so far in this chapter. It's going to be related to something to do with one of those 6 factors that are required for bacteria to grow: food, acidity, temperature, time, oxygen, and moisture. And if you remember, there was an acronym, FAT, TOM. Don't have to know it, but that's what that stands for. All the conditions that are required for bacteria and microorganisms to grow in food. When manufacturers package food, they are attempting to maintain a very high quality food product while also keeping the food safe for as long as possible. In a way, you are putting food in a bubble and barring entry to all items that can cause contamination of that food. There are chemical reactions that will occur inside of a food package, but these reactions can be significantly minimized when the right environmental factors are controlled. Remember Fat Tom. What do the O and the M stand for? O is for oxygen and M is for moisture. And water vapor, that could fall under moisture. These are two ingredients that are necessary for microorganisms to live and thrive in. By cutting these out, microorganisms simply cannot live. Some containers will contain moisture, and they will have a slightly shorter shelf life than products that are packaged without moisture. But if you look at many canned foods that have juice or water in them, they still have expiration dates years after their original production, which speaks volumes about what can be done to a food in the complete absence of just oxygen. Also, packages can provide barriers to both physical and chemical contaminants as well as microorganisms. By not allowing these items in and also controlling moisture and oxygen, the shelf life of foods can be significantly extended. There are multiple types of containers that we can put foods into, but I'd like to just highlight metal containers for two reasons. One, because they have an incredible ability to keep out microorganisms and prevent spoilage. And at the same time, they're also, under the right conditions, very susceptible to very significant health effects. So they are a strong barrier against chemical, biological, and physical contaminants. They keep foods in a relatively safe enclosure and very safe environment that's very difficult for outside factors to influence and for bacteria to grow and multiply to harmful levels. However, if metal containers are bent, some of these metal containers use corrosion-resistant materials, and this can cause little cracks in that corrosion-resistant material, which can allow that metal to come into contact with the food. And it's very possible that that contact just from those chemicals from the metals touching the food, that can cause a release of chemicals that can produce very undesirable reactions that can cause the food to spoil and become very dangerous for consumption. So be very careful with any bent aluminum or metal cans that contain food. Two of the more intensive methods of packaging involve aseptic packaging and modified atmospheric packaging. With aseptic packaging, we're talking about putting foods that have no bacteria in them in an environment that has no bacteria in it. To do this, we have to do two processes. We have to sterilize the food, and again, sterilization is different from pasteurization because pasteurization kills some bacteria, sterilization kills all of the bacteria. So we sterilize that food, sterilize the packaging, put the food in that packaging, seal the packaging, and so you have just created an environment that has absolutely no bacteria. So that food is going to have an incredibly long shelf life because there is no bacteria that is in there that can cause spoilage to occur. With modified atmospheric packaging, this is quite a bit different. This involves altering the gas that is surrounding the food so that the shelf life of that food is going to increase. So generally this involves putting the foods in the presence of less oxygen, and this makes it more difficult for bacteria to grow and also prevents against oxidation reactions that will reduce the quality of food. Now there are some foods that do benefit from being in the presence of oxygen, such as fruits and vegetables and red meats. And these foods may be packaged under those specific circumstances. But either way, with modified atmospheric packaging, you are modifying the amount and types of gas within the package or the container of the food so that the shelf life and the quality of food is going to either remain stable or increase.